All right, so we get started? All right, thanks. So my name is Nipan Patel, and I'm a faculty member in both MCB and IB, and I'm going to be teaching basically for the next 5 weeks, Bio 1A. And that's my email up there if you need to reach me. And then just like Dr. Dowd and I'll have office hours Monday and Friday, the times that are listed in the syllabus and up here on the screen in VLSP 2084. So this is the first time I've taught 1A, so I'm used to teaching more upper division courses in genetics and developmental biology. But I have a pretty broad background in biology, so this should be fun for me, I'm hoping. And so today, we're not going to try to cover too much. I mean, I know partly because you just took your exam, and so we'll go kind of slow today. And that'll also give me a feel for how much material I can cover in a single lecture. And then you've got the syllabus, and you've got some idea of the parts of the book that we're going to cover. But the book is really, for me, sort of background for you. And I can emphasize that what you'll see on the exam is what we cover in class. Okay, so for a lot of the topics we're going to talk about, and some things the book goes into way more detail than I plan on going into. So you don't need to worry so much about that. Okay, and we'll try to cover, in a few things in the lecture, we might go into a little bit more detail than the book. You'll also see that we're going out of order now in the book chapter. So in the first 5 weeks, you went pretty strictly in order through the book, learning about biomolecules, enzymes, metabolic pathways, and so on. And I'm following a template that was left for me by the person that taught this before and by the faculty member that teaches it in the fall semester. So I'm going to try to follow that template. It's a little bit different than the order of the book, so you'll see that we're skipping many chapters ahead, and then we're going to go backwards and forwards again. But you'll see that there's actually a sort of an intellectual reason for that. There's a strand of thought in that. So we're jumping actually right away to a chapter on viruses, but we're going to cover viruses very superficially today for a very particular reason, which you'll come to understand as the lectures go on. Let's see what else. So I'm also going to use the iClicker, though I'm not going to use it today, but in future lectures we'll do it, and we'll do it the same way that Dr. Doudna did it. A couple questions per lecture. I think it's half a point for answering, a half a point for getting it right, and Mike Meehan will take care of adding up those scores, but we'll use it as a way to sort of get feedback. I also tend to ask questions in my lectures, and I know it's kind of hard with this many hundred people in the room, but, and I'm sorry I'm not going to learn all your names, but if you just want to shout out answers, that's fine, okay? So we can have a little bit of a discussion as we go along. Okay, so what you already know, so based on what I've seen from the lectures that are posted there, you have a pretty good idea now about the structure and function of molecules that are associated with living things, such as proteins, lipids, carbohydrates, and nucleic acids. Okay, you understand or you have a basic outline of the structure and organization of a cell, and then the enzymes, and how the activity of those enzymes is regulated, what those enzymes do, and how they regulate in a general sense. There's obviously tens of thousands of enzymes, and you've just learned a couple examples so far. And then energetics, you've learned a little bit towards the end of the first 5 weeks about metabolism and photosynthesis. Okay, so what are we going to do now? So what the goal of these next 5 weeks is really, in a nutshell, it's teaching about introducing you to molecular biology and genetics. Okay, and the emphasis here is really going to be on DNA. So you've learned about a lot of biomolecules going so far. And you've learned a little bit about DNA, and you know DNA structure, you know a little bit about RNA. And I guess you got an introduction, and probably most of you had plenty of biology in high school too. So you know the concept of how DNA is used to template RNA, and that RNA is read in order to make a protein, right? So everyone's familiar with that general concept, right, of the flow of information. And if you're not, we're going to go over it, but I assume that that's something that most of you have seen plenty of times. But we're going to talk about this in a lot more detail now in these next 5 weeks. And so we'll talk about how that DNA is replicated, and we'll go into why this is an important step. And then for cells, once that DNA is replicated, there's also the process of cell division. We'll talk about that. But the biggest emphasis will be really mechanisms of inheritance. So how information is passed when a cell divides, so that both cells have that information, or when an organism reproduces, so its progeny have that information as well. Okay, but let's step back quite a bit, okay, and try to give you an idea of where we're coming from with these kind of questions that we're going to ask now, in a very basic sense. So start with the question: how do we know that something is alive? Okay, so you've got here a rock and a rockfish. Okay, and it's pretty obvious that the rock is not alive and the rockfish is alive. I think most people would answer that way. Okay, but does anyone want to try to tell me why you would answer that way? What's that? Okay, so one is reproduction. All right, so rocks don't really reproduce. You can break them, okay, but they don't really reproduce, right? Whereas a rockfish should be able to reproduce and have progeny. Anyone else want to shout out any other things? What else would tell you that one is alive and the other's not? Okay, one has metabolism, so one actually uses energy, right? And so in the case of something like a rockfish, it's eating food, right? And that's what it's using to stay alive, so it's using that as a source of energy, whatever it eats. And then you learn about plants, so plants don't have to eat anything, but they use sunlight as a source of energy in order to fix carbon, and so they can stay alive. Okay, any other things that would distinguish one from, yeah, they interact with their environment, and they respond to their environment in various ways, right? So like a rockfish, obviously, if you disturb it, it moves. A plant doesn't move, but it does respond to its environment, right? So as the light moves, right, it moves its leaves to follow the light and so on, so it has a response to the environment, whereas, the rock is an inanimate object, so it doesn't respond. Any other ideas? Yeah, so over time, the rockfish population can change. Okay, so you might imagine that this rockfish has a certain color to it because that's the color of the rocks that it lives on. And so one might think that if you ended up having this population in a very different environment, that the color of these rockfish might become different over time. Okay, and so that's something else that living organisms do, is that they evolve over time. Okay, so just to go over those, right? So what you guys answered. So reproduction, right? So a very obvious thing that living organisms make copies of themselves. All right, they also have order to them, right? So you generally see patterns in living organisms, and those patterns are repeated, right? So even simple things, like we'll talk about bacteria. Bacteria usually have some sort of particular shape, whether it be like a rod or a sphere or a little spiral or something like that. And as they divide, they tend to keep that shape. Okay, and then of course other animals and plants you think of have very complex shapes, and their progeny have those shapes as well. Okay, and then there's energy processing metabolism. So there's an energy input into living systems in order to maintain them. So there's an elaborate system in all living systems to maintain that energy. All right, and then finally there was the adaptation and evolution, right? So in this case you've got this little seahorse that's matches perfectly into the coral that it's hanging on to, and that's an exquisite example of the adaptation that organisms have over time. This may take a very, very long time, more than we can usually observe if you're looking at large animals. They evolve. If you're looking at very small things like bacteria, which have very fast generation times, you can actually see rapid evolution. So what's an example of how a bacterium would evolve? Something that's very simple but clearly shows adaptation to an environmental change. Yeah, so that's a classic one, right? So you can have antibiotic resistance. So you could be sick with something, you take some sort of antibiotic, it seems to be working, and then suddenly the infection will flare back up. Okay, and that's usually because somehow the bacteria become resistant to that. And in fact, we'll talk about the mechanism of that resistance, okay, how that happens. And that's an example of altering the genotype and the phenotype of an organism, right? Okay, so then as we understand it right now, life, living things are divided into 3 large domains, okay? There's the domain that's called bacteria, the domain called archaea, and then there's the eukaryotes, the eukarya, okay? So the difference between eukarya and these two together are called the prokaryotes, right, is the presence of a membrane around a membrane-enclosed nucleus, okay? So the DNA is within that structure. So you study the structure of a cell already. You know that in something like one of your cells, there's a nucleus, and that's a membrane-enclosed organelle, and all of the DNA is inside of that. Okay? In bacteria and archaea, there's not that organized nuclear envelope around them. Okay? That's one of the differences that you find. Okay? And in general, of course, the bacteria and the archaea are small, and then these tend to be more complicated organisms, so that's not always true. We'll talk about that in a second. So it used to be that bacteria were just thought to be bacteria, right? That there was no distinction into these 2 giant, what we call now kingdoms, or domains. And this was a relatively recent discovery in the last few decades, that in fact there was a whole group of bacteria, what had been known as bacteria, that were very, very distant from other bacteria, and that you could divide bacteria, what then had been called bacteria, into 2 giant groups that were very distantly related to each other. And in fact, there's a thought that these archaea bacteria are actually slightly more, or a little bit more closely related to everything, the animals and plants, the eukaryotes, relative to the sort of true bacteria. Yeah. Yeah. It's a little hard to prove. Okay? It's also very hard to prove because there's not a 4th domain of life as what we call an outgroup to compare it to. Okay? And so there's a few things about the biochemistry of archaea that people have used to argue that they're closer to eukaryotes. But you don't need to worry about that relationship necessarily, just to know that these are very different types of unicellular organisms. Okay? So then, and they tend not to have, obviously, they don't look very complicated to you, right? Because they're single cells, and so they can't have too many different shapes to them. Okay? And of course, we tend to ignore them in many ways because we don't see them very much, right? So even the largest bacteria would be barely visible to a naked eye. In fact, most bacteria in archaea you need a microscope to see. But in many of them we can culture, but there's lots and lots of them that in fact we can't culture. They're out there in the world doing things and living, but they won't grow in agar plates and things like that. So we don't really know how to grow them. So we know from DNA sequencing that if we go into the ocean, that there's in fact enormous numbers of these things living there that we don't know what they are, but they're there and they're reproducing, they're living their lives in some way. Okay, so but in the eukaryotes we have the organisms you're more used to thinking about. They're the ones that you see out your window every day, and they're divided up into a couple large groups as well that you'll be familiar with. So there's the plants and the animals, and these are 2 independent evolutionary experiments in multicellularity, right? In having cells stay together and form more complicated organisms. So they've arisen completely independently from unicellular ancestors. Okay. And then you have the fungi, okay, so mushrooms and yeast and things like that. And again, something like a mushroom is a bit larger, so you'll be familiar with that, whereas yeast is something that you don't really recognize. I mean, you know what it does, but you don't really think of it as an organism. Okay. And then there's protists, which is a very unfair characterization. So in fact, many scientists would be really annoyed knowing that there's a textbook that calls it just protists, because these are as diverse as any of these are. Okay. And some of these are individual protists may be more related to one of these groups than the other. Okay. So there's many lineages of unicellular organisms that are eukaryotes, so that they're unicellular, but they have a membrane around their nucleus. Okay. And so, for example, there's a unicellular organism called the Quiniflagellate, which is the closest ancestor to animals. Okay. But that's not the closest ancestor to multicellular plant. There's a closer unicellular ancestor that led to multicellular plants. Okay. And that unicellular ancestor that led to multicellular plants already had chloroplasts in it. Okay. So and then that led to the plant kingdom. So this is actually a huge group of organisms. All right. Okay. So go back to these bacterial type things, so the bacteria and the archaea. And so they are, of course, very ancient too, right? So if we think about it, the Earth is something like 4.6 billion years old. And it's not that for about the first billion years there really wasn't anything alive on the Earth. Okay? Conditions weren't actually favorable and life hadn't actually come into existence yet. There were no replicating biomolecules or anything like that. And then life appeared about 3.6 billion years ago. Okay? But for about 2.5, 2.6 billion years, that life was always just unicellular. And there were no multicellular organisms. So the world was in fact dominated by single-cell organisms that were probably something like present-day archaea and bacteria. Okay? And then the Cambrian explosion, which probably all have heard about, right, when animals really erupted into lots of fantastic body forms, that occurred about 550 million years ago. Mammals didn't appear until about 200 million years ago, and anatomically modern humans only about 200,000 years ago. So most of the history of life is really the history of unicellular organisms. And the things that we see now are really latecomers onto the scene. Okay? And so these tend to be much more recent, and these are very ancient lineages, right? Not that, keep in mind that anything that's alive now, right, it's the same ancient, it's been a continuous lineage of living organisms to the present, no matter what animal or plant or microbe you are. Okay, you still trace back to an ancestor that's billions of years old. It's the common ancestor. So just to get around the idea that it's not really fair to say that bacteria are primitive, right? They're every bit as advanced as a human is. They just don't look it to you. Okay, but they've been around and they've been evolving. And in fact, if I ask you the question, what organisms are the most complex, what would you say? Just even naively. What's that? Why is that? Okay, and that's fair enough, because the word complexity, you have to define it. So some people would say, well, like humans are the most complex. We have the most complex brain. We think, we have consciousness and things like that. That makes us more complex. But that's a very human bias that we, of course, want ourselves to be the sort of pinnacle of everything. And so we define ourselves as being the most complex. All right? But it's a real shortcoming. So one of the nice illustrations of this, we think of, like, say, a fruit fly as being a very simple organism. Okay? But in its world, it's not such a simple organism. So if you look at fluorescent lights, right, our mind works too slowly to see that the light is going on and off. Right? So a fluorescent light is going on and off at 60 hertz. But our brain works too slowly to see that. But a fly's brain works so fast, it sees the lights go on and off. Okay? And that's because its mind has to work much faster to fly. And so it does that. And so in its own way, it's very complex. But it turns out that if you think about metabolism, the most complex organisms are actually these, the bacteria and the archaea. They can do way more things than any eukaryote can do in terms of metabolism. So we're pretty stuck on a few ways to get food, to get energy. Whereas the prokaryotes have explored all sorts of ways to get energy. So not only can they do the typical things like photosynthesis, right, just like a plant can, but they can use other energy sources. They can actually use crazy energy sources like other chemical reactions in order to fix carbon and to carry out their metabolism. So that means that they can live in really unusual environments. And so, in fact, archaea were discovered because a lot of them live under really extreme conditions. For a while, it was thought that, well, that was a defining characteristic of the archaea. It's not. They can live in very very normal environments, and some sort of now what are called in the bacteria can live in extreme environments. But this is just an example. This is the prismatic pool in Yellowstone, right? So it's this sort of boiling cauldron of water. It's not only very hot, but it's very high concentration of all sorts of nasty minerals and everything. And yet there are bacteria, there are archaea and bacteria that live perfectly fine in that environment. So there are unicellular organisms, prokaryotes, that can live in conditions above boiling. Okay, so they have adapted to those kinds of conditions. They can live in extremely high salt conditions. They can do things like use what we would consider toxins like formaldehyde as a carbon source. We're perfectly fine with that. So in fact, most of the diversity actually exists in the archaea and the bacteria in terms of metabolism. But if we think about morphology and shapes of organisms, then it is the eukaryotes that we think of as having complexity. Okay, so now let's get back to the sort of a central question that we're going to start delving into. So we've sort of talked about living things and some of their properties, and we said that one of them was their ability to reproduce. Okay, but a key question we can ask is, when organisms reproduce, why do the offspring look like the parents? Okay, was that? Well, we'll get to that, right? Okay, so I mean, most of you know the answer to some level already, right? So why is it that the baby giraffe looks like its parents? And then the same thing with like even a bacterial cell. So when a bacterial cell divides into two, the two daughters look alike. Okay, and then they keep dividing, and so you can have millions of offspring or millions of bacteria from a single one after not very long, since they divide every 20 minutes or so, right? So they grow very quickly. Yet they all continue to look more or less the same. So why is that? And it's not just that they look the same. Of course, think about it. They're full of all sorts of molecules. So you studied a lot of the things that are in a given cell. So you have sort of complicated lipids, you have the RNA and DNA, you have all these proteins. A bacterial cell probably has a couple thousand proteins in it. So how do you keep making more and more of those so that each cell looks the same? And that's really going to be kind of a central question that we're going to tackle in the next few weeks. So what are the instructions, okay, that allow an organism to replicate itself? Okay, and how does that process work? How do you make a faithful copy? And then we'll talk about how you can actually alter the copies as you go. How does that work? All right. So someone already said, well, it's DNA. Okay, so the question is, how do we know that? How do we know that DNA is the material that carries the instructions for an organism? Okay, so what we'll do is we'll talk about a few experiments right now that indicated that in fact DNA was the molecule that gave rise, that's responsible for inherited patterns. And you have to remember that 80 years ago or so, no one had any idea what the molecule would be. So they knew that cells were composed of a lot of large biomolecules. They knew about proteins and lipids and carbohydrates and nucleic acids. But there was a lot of debate as to what the material was that actually carried the genetic information. Okay? And in fact, many people for a long time thought it was proteins that actually carried that information. And so let's look at a couple experiments that get to this. So this is one done in 1928 by this guy, Frederick Griffith. And so what he was looking at is a bacterium, a pneumococcus, that causes pneumonia. And in this case, it works in mice as well, so he could use experiments in mice. And he noticed that there were two strains of the same bacteria, and they called them S and R. And strain S, okay, when you injected it into a mouse, it would kill the mouse. Okay, but strain R, when you injected that, the mouse stayed healthy. Okay, and then they also noticed, and the reason they were called S and R was that these cells looked smooth and these cells looked rough. Okay, and the reason these ones were smooth was because they had a coating on the outside of them, okay, a polysaccharide coating. And the idea was that that's what was allowing the bacteria to actually infect the cells and to kill the animal. All right? So both of them could live in a proper culture medium, maybe fine, but in terms of infection, this is the strain that could. But they were the same bacteria, okay? And it was just this correlation with this morphology that indicated that one was actually going to cause pneumonia and the other one wasn't going to actually cause death in the mouse. All right? So one experiment that he did, of course, was not so complicated. He could heat these cells up, and they would die. All right? And then you would inject them and nothing would happen to the mouse. Okay? No big deal, because you heat things up and they die. Even bacteria, most bacteria will die when you heat them up high enough. Okay? And then you inject them in and they're harmless, right? In terms of actually being able to create an infection. Okay? It doesn't work if the bacteria is made of toxin and toxin's resistant to heat. That can happen. But in this case, the bacteria would have to infect and replicate, make more copies in order to cause pneumonia. And if they're heated, that's not going to happen. Okay? But then he made an interesting observation. So if he heat killed the S bacteria and then he mixed them with live R cells, okay? Mixed this together and injected it, now the mouse would die. Okay? So give me a reason that would happen, an idea that you might come up with to explain that observation, that the dead S bacteria when mixed with live R could still kill the mouse. Okay, but the S cells are dead. So what would be the nature of that interaction? Yeah. Okay, so the basic idea is that's what they latched onto, was that somehow the R cells, right, were getting information from these dead S cells. And that one idea was that was genetic information. It was information on how to make this polysaccharide, this coat on the outside. Okay? The other possibility you could think of is that in fact what happens is these R cells just directly use that polysaccharide coat. Okay? And then they use that to infect, and then they can start the infection. But in fact we know that that's not happening, because after the mouse dies, you can get out live S cells. Okay? So even though these have been dead S cells and live R cells, the infected mouse now produce live S cells. Okay? So the idea was that there had been a transfer of genetic information, of some sort of information molecule that now allowed these cells to be able to make that coat and to become infectious. Okay? So it's a very early experiment, and of course you've already started to say things like, well, conjugation and DNA and stuff. But remember, they didn't know any of those. They didn't know that those were the molecules responsible for information, for the basis of genes and things like that. Okay? But that's sort of an initial observation. And so we'll go through another experiment, which will get to the fact that it is DNA that's actually being transferred. Okay? We'll come back to this example later on in the class. I mean, not today, but later on, and tell you exactly how it's happening. Okay? And it has a lot to do with the same sort of phenomenon of antibiotic resistance and things like that. So the other way that people saw this was studying viruses. So this is a tobacco